Hello, and welcome to ACES with Sarah, Alicia, Evelyn, and Cassidy. We are here to help you ace your next biology quiz. Today's topics involve photosynthesis and cellular respiration. Although photosynthesis and cellular respiration are completely different processes, they do have some similarities. Today we're going to cover both of those processes, highlighting their differences as well as their similarities.
Photosynthesis, unbalanced and balanced equation.
Although photosynthesis and cellular respiration are completely different processes, they do have some similarities. Today we are going to cover both of these processes, highlighting their differences as well as their similarities. First, for many, including myself, photosynthesis is taught as the process in which carbon dioxide is taken in by plants and releases oxygen into the atmosphere. From there, humans breathe in the oxygen, and the cycle restarts again as humans breathe out carbon dioxide. This understanding is true but very broad. Have you ever wondered what actually happens specifically during the process of photosynthesis, or how the water and carbon dioxide react to produce glucose or oxygen? The first stage of photosynthesis is the light-dependent reaction. As you may think of dependent, you may think of something relying on an external force or variable. Ultimately, this is exactly what the light-dependent reactions are, and the factor that the light-dependent reactions are dependent on is light energy. In the light-dependent reaction, the light energy is exerted on the chloroplasts. The chloroplasts are located within a plant cell that undergoes photosynthesis through absorbing sunlight. Within the chloroplast, there is a structure that you will need to understand to see the scale of the process of photosynthesis. In the chloroplast, there are many stacks of little discs known as granums. An individual disc is called a thylakoid. Inside these granums, there are electrons. When the sunlight is absorbed, these electrons become excited and move around very quickly. An electron is released along with energy known as NADPH. In the mitochondria, the energy-making part of animal and plant cells, an energy known as ATP is released. Light-dependent reactions convert light energy to chemical energy that will be used in the light-independent reactions. Water is used to replenish electrons in the chloroplast. This compound is consisted of 2 hydrogen molecules and 1 oxygen molecule. The molecules float around in the cell. The hydrogens split off and help replenish missing electrons. The oxygen molecule bonds with other oxygens to give off the oxygen we need to breathe.
The next stage is the independent stage. In this stage, light is not required for the reaction to happen. It is also known as the Calvin cycle. This stage relies on NADPH and ATP and the O2 released during the dependent stage. This stage uses chemical energy to convert carbon dioxide into glucose known as sugar. After energy is converted to the ATP and NADPH convert back to ADP and NADP+, because the electrons are used during this stage. An interesting fact is that something were to happen to the plant, it would be able to survive a bit longer than the plant that completed the light-dependent reactions. Also, this stage takes place in the stroma, which is the fluid area outside of the thylakoid membrane. An analogy would be, since ATP and NADPH are constantly getting renewed during this process, the dependent and independent stage are constantly repeating, similar to a battery.
Venilaria was mainly used by farmers who use it to kill broadleaf weeds and clover. The reason why it is used in the first place is because when farming there seems to be many weeds growing along the side of the crops that are planted. When the weeds grow, they are limiting the crops' resources that they need to grow, like the fertilizer and water. Farmers use herbicides like Venilaria because it helps the soil structure gain the ability to hold water better and earthworms to develop the soil. Using this herbicide is considered as much better advantage for farmers compared to plowing their unnecessary weeds growing in their crops. Venilaria herbicides are photosynthesis inhibitors. When Venilaria inhibits the plant, it slowly kills the entire thing. The plant immediately shuts down the process where the plant is producing food binding to specific sites within the plant's chloroplast. This happens because it blocks the flow of electrons in the photosystem to bind protein in the thylakoid membrane. Venilaria inhibits the reaction which ATP and NADPH plus forms, which causes the photosynthesis process to damage and causes permanent lack of food production for the plant. The moment the process shuts down, the plant begins to starve in a slow pace. The symptoms coming from this treatment include leaves becoming yellow, beginning to die, and dying at the end. Cellular respiration has three cycles: glycolysis, Krebs cycle, and oxidative phosphorylation. Glycolysis is a breakdown of glucose. This process takes place outside of the mitochondria and in the cytosol. There are 10 steps in glycolysis in which many different enzymes play important roles in breaking down the different glucose molecules. Oxidation is the way molecules share electrons. NAD plus is a coenzyme which uses its role to transport electrons, and when NAD plus picks up an electron for transport, it soon becomes NADH, which takes part as an electron donor. Phosphorylation begins to occur when enzymes act to move phosphate groups around. When a phosphate group is added to an ADP, it transforms into ATP molecule. Glucose is a 6-carbon molecule which contains hydrogen and oxygen as well. The first step occurs when an enzyme called hexokinase acts on the glucose molecule to add 2 phosphorus molecules. The result from that is a 6-carbon sugar molecule called fructose. Phosphates have been added, meaning that it will continue to move around while the enzymes continue to act upon the glucose molecule, which uses 2 ATPs. Then comes an enzyme which splits a 6-carbon molecule into 2 3-carbon molecules. When active, they will react differently to the enzymes that continue to break down the glucose molecule. The NAD plus becomes NADH because it comes in to rearrange the way 3-carbon molecules share electrons. From there, 4 ATPs, 2 pyruvate, and 2 NADH are formed by every glucose that has broken down in glycolysis.
The second stage is the Krebs cycle. After the glycolysis stage broke down glucose into 2-pyruvate, they are converted into 2-acetyl-CoA. To make this demonstration easier, we're only going to use 1-acetyl-CoA. So through the process, 1-acetyl-CoA enters and converts ADP into 1-ATP, and 3-NAD plus into 3-NADH. And lastly, 1-FAD plus into 1-FADH2 when electrons are added through the process. Also through this process, 3 carbon dioxide are released. This is important to note that in the diagram we only used 1-acetyl-CoA, but if we had used both, they would have doubled. So in the end, you would have 6-NADH, 2-FADH2, and 2-ATP. Lastly, this process takes place in the mitochondrial matrix, which is the empty space in the mitochondria.
After the Krebs cycle, the oxidative phosphorylation process takes place. This occurs in the intermembrane. Alike to the cell membrane, the mitochondria has a phospholipid bilayer not once, but twice. There is an inner and outer membrane, but the oxidative phosphorylation takes place in the intermembrane, which is the space between the outer membrane and inner membrane of the mitochondria. During this process, FADH2 and NADH, the electron carriers created in earlier processes, bring electrons to the protein channels, but they are later emitted as hydrogen molecules to the inner membrane, creating a large amount of hydrogen on the inner membrane and a lower amount on the other side. Naturally, the molecules in larger amounts want to move to the other side because molecules move from high to low concentration. But how does this occur? It occurs by transporting through the ATP synthase. When they move through the ATP synthase, the kinetic energy is transferred into chemical energy, which ends up bonding with ADP to create ATP. Also, the extra electron from the protein complex bonds with 2 hydrogens and an oxygen to create water. Rotenone is an insecticide and pesticide found in plants such as jicama and in the root family Fabaceae. This family is known as the bean or pea family. Rotenone is also a pesticide, meaning that it kills fish. This plant can also be used to treat scabies and head lice. The poison is not very toxic to humans or mammals, but is towards fish and insects. This poison is easily absorbed through the gills, but not as well as if it's through the GI tract or the skin. People rarely die from rotenone because they will vomit it up, but if they ingest it, they will most likely die. A study was done at the beginning of the 20th century using rats. Scientists gave the rats a dose of rotenone and found that the rats were experiencing symptoms that were very similar to Parkinson's disease. Rotenone has been used to kill fish for years, once in lakes and rivers. Scientists also found that farmer workers exposed to this pesticide had symptoms of Parkinson's disease.
We have come to an end. Today you learned about photosynthesis and cellular respiration. If you saw this video, you might get an A for your next assignment. Thank you for watching. This comes to you from ACES.